All right, I'm going to go ahead and get started. We have a class coming in here right at 1 o'clock. So it would be very easy for me to give a very long introduction for Carol, but I'm going to keep my introduction relatively brief to make sure that she has plenty of time to share her research. It's my pleasure to get to introduce our distinguished speaker for today, Dr. Carol Barnes. She is currently running her research group and has for many years at the University of Arizona, where she wears a number of different very prestigious hats. So those include the Regents Professor of Psychology, Neurology, and Neuroscience, the Evelyn F. McKnight Chair for Learning and Memory and Aging. She's also the director of the Evelyn F. McKnight Brain Institute that's at University of Arizona, and is the director of the Division of Neural Systems, Memory, and Aging. Carol started her training with undergraduate work at the University of California in Riverside. She then moved on to get both her master's and PhD in psychology at Carleton University in Ottawa. And then it sounds like she really went all over the world to get some of the cutting edge training during postdoctoral work in discoveries about plasticity, synaptic plasticity that was really just coming out, LTP, how does this work, and being able to learn cutting edge techniques that were used to investigate this phenomenon, slice electrophysiology and single unit recording. So that took her to the University of Dalhousie in Nova Scotia. I'm looking at Anne and Carol. Dalhousie, dang it. All right, I tried. You'd also at the University of Oslo, and she was also at University of College London. So really going around the world chasing these early discoveries about what might be some of the cellular substrates of learning and memory. Carol started her lab at University of Colorado, Boulder, and was there for a while before moving her group to University of Arizona. And at University of Arizona and throughout her career, her research group has really been dedicated to the goal of understanding how the brain changes during normative aging. They've been going after some questions about what are the molecular, cellular, and circuit level changes that accompany aging? And then what do the consequences of these changes end up being for information processing and memory? What are the types of changes that determine inter-individual differences in whether or not cognition is preserved during aging, or whether or not the system starts to tip toward dementia and other cognitive challenges of aging? So in terms of honors and awards and just accomplishments throughout her research career, I could list many, many things, but the things that really just I find incredibly impressive are just her sheer productivity of her and creativity of her research program. Carol's group has produced over 200 peer-reviewed publications. Many of her trainees have gone on to start their own research groups, including some faculty who are here at UCLA. She's a member of the National Academy of Sciences, a former president for the Society for Neuroscience, and has won a number of awards, including the Gerard Prize in Neuroscience and the American Psychological Association Award for Distinguished Scientific Contributions. Two more quick statements, and then I'm turning it over. If you've heard of the Barnes Maze, this is a creation of Carol Barnes to help us really have more tools for understanding memory and cognition. And then I just wanted to share a tidbit. I learned this morning that also in her personal life, Carol is a trailblazer, not just being a pioneer in the research world, but she has also created a multi-mile hiking trail in the mountains behind her home in Arizona. So a trailblazer both in science and in her personal life. It's such a pleasure to have you here and looking forward to hearing about your work. Please join me in welcoming.
Thank you so much for that wonderful and warm introduction. It's really a pleasure to be here for your CTSI speaker series. And it's great to see old friends and colleagues as well around the room here. Some of you may know that my research has focused on the understanding of brain mechanisms of memory changes with aging. And since very, very early days, since the beginning in the mid-1970s, before the National Institute on Aging actually existed. And I started with rats as my experimental model of aging back then. And in the early 2000s, I expanded my lab to include non-human primates, a bridge closer to humans. And in the past year, in fact, I've extended my research program in humans for two humans for the first time, and have been awarded a grant to fund an effort that we call the Precision Aging Network. And let's see here. Yeah. Right. Oops. There. All right. We've got this down. One of the projects of the Precision Aging Network is to collect cognitive health and lifestyle survey data from the largest geographically representative sample of individuals from 18 to over 90 years of age across the United States. We currently have over 350 participants using the MindCrowd web-based testing platform, and we aim to expand this cohort. Each blue dot that you see here is a test taker in our MindCrowd test. A second project is to collect data from 4 geographic regions in the United States, Tucson, Miami, Baltimore, and Atlanta. You see those as the 4 stars on the map. And these sites will enrich their enrollment for in-depth laboratory analyses of targeted race ethnicities. There are 2 other projects that I don't have time to go into now, but the overall goal is to define normative cognitive aging profiles across age, socioeconomic status, geography, race, ethnicity, so that we can eventually have a scientific basis for developing individualized interventions to optimize brain and cognitive health. Now, I'm not going to talk about any of this today. I just wanted you to be aware of this, and in case anyone is interested in speaking to me more about this after the talk. I want to, of course, encourage you all to take the MindCrowd test at this website. If you're over 18 years of age, that would be great. You'd be contributing to this US-wide effort. Okay, the overall rationale for the type of basic animal work that I've done for most of my career stems from the idea that if you understand the basic biological mechanisms of memory and how it's altered by the aging process, the normative aging process, then this is going to enable you to identify or distinguish memory changes that reflect disease states from those that are independent of disease. And as you probably know, it's only humans that are stricken with Alzheimer's disease. Other animals do not spontaneously contract AD. And so what I'm studying in the rats and monkeys that I'll be talking about today is the normative aging process. Now, one advantage to having been interested in aging and memory for almost 5 decades is that it reminds me that we've truly come a long way in separating our assumptions and myths about the aging brain from actual knowledge about how memory traces are laid down in the brain, in the connections between cells, and how these memory circuits are changed over the course of aging. And perhaps this is better discussed, you know, in a more casual environment, maybe over a glass of wine or something. But to me, aging is not a disease. It's a process that occurs in all of us across the lifespan. Furthermore, aging does not just happen to those of us who can study it, but it happens to all species, all animals. And so we can learn a great deal about the fundamental principles of the aging brain and cognition by studying animals other than humans, who also show these brain and memory changes as we age. Now, interestingly, these memory changes happen at the end of the lifespan of whatever species you're studying. That is, for a rat, is about 2 years old in the species that I study. In monkeys, it's 22 years old. You multiply by 3 to get human equivalent years for monkeys. And transition to old is considered to be 65 years in humans, but I think that's far too young. I think we should be extending it out now that I'm well past this. So what this means then is that there's a fundamental biological process that occurs in the tissues of the body, including the brain, that is essentially sped up in rats and monkeys compared to humans. And because I'm interested in translating my neurobiological findings to humans, if possible, I've used behavioral methods that can be tested similarly across species. Of course, you can't have language in these tests, and also that are well established to change with age in all species that are tested. So for focus here and to constrain the information that I present to you about brain aging, I'm going to emphasize the hippocampus and spatial memory as essentially an example system to study change with age in all species. So I'm going to emphasize the fact that there are clear region-specific differences in the impact of aging, for example, on synapse function. And I know that this particular audience is very diverse with respect to background. And so what I hope to do is give everybody maybe just a little bit of something that they might be interested in as we go along in this talk. So I'll begin with cognition, and a number of tests that have been used to show hippocampal dependent memory changes with aging, and spatial memory impairments can be demonstrated in old rodents in this circular platform task, and also in the Morris water maze. These tasks are conceptually identical. The animal has to remember where escape is possible, and either onto a platform that's hidden beneath the surface, as you see in the lower right, or as you see here, into a dark escape box underneath the platform's surface. And not only do old rodents show this type of hippocampus-dependent spatial memory deficit, in fact, old primates, both monkeys and humans, also show spatial memory deficits. Now, Peter Rapp's data are consistent with older monkeys in an open field foraging space, showing poorer spatial strategies than younger monkeys. And one of many human experiments I can show you, is a dry version of the Morris water maze in older humans, where they also perform more poorly than younger individuals. And just the point that I'm trying to get across here is that spatial memory problems that implicate the hippocampus exist at whatever age and a species is considered to be old, as I mentioned in the beginning of my talk. So a number of factors may contribute to these observed memory changes. And I want to begin to discuss this with this slide, showing you a summary of what was thought to happen to the aging neuron way back when I started to be interested in memory in the early 1970s. These are entorhinal cortical neurons. You see the beautiful neuron in the younger 20-year-old individual. And by the time you get to the 90-year-old cell, the cell would be 90-year-old person, the cell would be quite shriveled. And of course, this is particularly disturbing depiction of of brain aging, because as you know, your neurons are largely born before your birth and die with you at the end of your life, with the exception of neurogenesis. But we now know that this depiction of brain aging is wrong. In fact, for the hippocampus, there is no cell loss, not in the granule cells, C3 or C1 pyramidal cells across age, not in rats, mice, dogs, monkeys, or humans, where these cells have been stereologically counted. And more good news is that the biophysical properties of hippocampal cells are also quite intact. The old cells are quite healthy. And if you record from these single cells in hippocampal slices, as listed here, you see that most membrane properties are similar in most respects in old cells compared to young cells. So you might say, well, okay, if in the hippocampus there's little cell loss and the cells that remain are healthy, what is normal brain aging then? And why do we observe spatial memory changes with age? Well, there are a number of reasons. Many properties of hippocampal cells do remain similar, just as I've described for you. Most biophysical properties are similar, but there is one group of findings in aged rats where changes do occur essentially uniformly in all three hippocampus subfields. And the two examples that I want to discuss with you include declines in the amplitude of the cholinergic slow EPSB and increases in gap junctional connectivity. So the first, I'll talk about the cholinergic slow EPSB. When cholinergic fibers are stimulated at particular frequencies, a slow muscarinic response, as you see here to the left, can be elicited in all three hippocampal subfields. And this slow EPSB is blocked by atropine, as you see on the right. So cholinergic transmission, which is measured as the amplitude of this slow EPSB, is reduced by about 50% in each of the hippocampus subfields. And this decreased cholinergic drive has the potential to reduce levels of excitability in these hippocampus cells. Okay. The second change that I want to show you that's observed in each hippocampus subfield in old rats is a change in gap junctional connectivity. As you're aware, gap junctions are these small channels made up of connexins that exist between cells, including cells in the hippocampus. And these channels have low resistance to the passage of electrical current. So fluorescent dyes can be injected into a cell and they pass through these gap junctions, but don't pass through synaptic junctions. And so by impaling one cell and filling it, you can count the number of cells that fill with dye and see how many cells are connected together and estimate the extent of coupling. And Gita Rao and I performed about 500 injections of carboxyfluorescein in young and old animals in granule cells, CA3 and CA1 pyramidal cells. And we found that a statistically significant increase in gap junctional dye coupling in old animals in each region by the percents you see here. And it was fairly equivalent across regions, reminiscent of the uniform change in cholinergic transmission. Now, one possible effect of this increased coupling could be greater excitability in the connected cell populations, because the cells with the lowest thresholds would tend to activate all the cells that it was connected to in the circuit. And one question that might be raised in this at this point is whether the reduced excitatory drive produced by reduced cholinergic transmission might be balanced by an increased electrotonic coupling, thus preserving overall function. And an experiment that I haven't been able to talk any student of mine into, but I wanted to do for years, is that there are ways to pharmacologically block electrotonic conduction. And so if increased gap junctional connectivity was a compensatory or an adaptive process, if you gave such a blockade, the behavior of old animals might actually be worse with such a treatment. And so someday I'll talk somebody into doing this, but it's an interesting possibility. Okay. All right. So I've shown you changes that occur essentially uniformly, but those are the only two I could kind of pull out of the hat for you. In fact, the majority, the changes in cholinergic and gap junctional connectivity, these are pretty rare. The majority of age-related hippocampus changes are remarkably subregion and input selective. And so just to review the cell types and the dendritic responses or specific inputs have been characterized. And for those of you who don't eat hippocampus for lunch and don't know all these connections, I just want to point you to what synaptic connections we are going to be talking about as I go through here. For granule cells, age comparisons of synapse counts have been made in the inner and middle molecular layers for CA3, in the stratum radiatum, CA3, both in the stratum lucidum and stratum radiatum, and in CA1 and in the stratum radiatum. Okay. Region and synapse selective changes have been observed in old rats and tend to fall into two general categories. And so I'm going to talk about these two categories of synaptic change. And the first is synaptic connectivity. The second, changes in synaptic plasticity. So starting with connectivity, there are clear changes with age in the connections made between cells, but the type of change remarkably depends on the synapse you're looking at within the hippocampus, even synapses close together. So beginning with granule cells, there are fewer actual synapses in both the inner and middle molecular layers. That is, the old granule cells have fewer perforated axis spinous synapses. And these perforated synapses are those implicated in synaptic structural changes following LTP induction. Now, interestingly, for the granule cells, while there are fewer synaptic contacts in the middle molecular layer, these are the medial interrhinal cortical projections, the synapses that remain in that middle molecular layer from this projection are individually more powerful in old rats. That is, when depolarization of a granule cell is measured following stimulation of a single interrhinal cortex axon, the resulting depolarization measured by the unitary EPSB is larger at old synapses. And we've interpreted this as possibly a compensatory response to the loss of synapses in this region by the aging brain, and have suggested that maybe this could be an example of the brain compensating at the synaptic level, or synaptic compensation. Now, frustratingly for some of us, the story is very different for CA1 pyramidal cells. For synapses in the stratum radiatum, this is the Schaffer collateral projection from CA3 to CA1. There's no change in the number of synapses across age. And Dan Nicholson has found a reduction in the size of the postsynaptic density at a subset of these CA1 synapses. We have shown that this unitary EPSB size is not different between age groups in CA1. So the strength of individual young and old synapses are the same. So if all old synapses are the same strength as are young, and you stimulate a group of synapses, you would expect the response size to be similar overall. But the observation is that the overall response is smaller in older rats, suggesting that while most of the synapses do not change their synaptic strength, there is a group of synapses that are silent in this region. And we speculated that perhaps the synapses with the smaller postsynaptic density might be those that are silent. And this is an interesting potential therapeutic target, because the synapses are there. Maybe you could turn them on somehow, and maybe that would be effective. All right, let's look at CA3. And this is unfortunately even more complex. So the granule cells send their mossy fiber axons into the proximal dendrites of CA3, the stratum lucidum. And these synapses there are fewer mossy fiber synapses in old memory impaired rats. Okay, there's a loss of AMPA receptors, and there's a weaker mossy fiber synaptic response. But right next door in the stratum radiatum, there are twice as many perforated synapses in this region in aged memory impaired young and memory unimpaired old rats. And this is it's the first report of an actual increase in the number of synapses in the aged brain. Likewise, there's an increase in AMPA receptors as well at perforated synapses in this region in impaired old rats. These data are pretty complicated, but what can be said for sure then is that CA3 in memory impaired old rats, it's going to be less influenced by input coming from the granule cells, fewer mossy fiber synapses, and more influenced by synapses that originate within CA3 itself. So it's talking to itself more than it is receiving input from the outside. The bus paper is fairly new, and I want to see it replicated and so forth. But it's really remarkable that actually we have the full gamut of synapse change, from loss to silent to more. So you pick it. So it's remarkable. All right. There's one other region specific difference in the impact of age and differing pyramidal cell types. This has to do with awake, freely behaving monitoring of pyramidal cell firing rates. And in the rat, there's no age-related firing rate difference in CA1 pyramidal cells, but in CA3, the older rats show increased firing rates, which was shown some time ago by Ian Wilson. Now, there's a new paper from Jim Kinnearan's lab as well that suggests that this increase may be localized to only a subset of CA3 inputs, CA3C, which is a bit of new complexity. But interestingly, actually recording from CA3 in old monkeys also show increased elevated firing rates of CA3 cells. Now, for rats, it's been shown that the number of neurons in CA3 does not change with age. I've already told you that. But the number of cells that express glutamic acid decarboxylase, or GAD, does decline. So these results suggest that GABAergic neurons switch phenotypes to become non-GABAergic neurons in CA3 rather than die with aging. Quite interesting. And we made an analogous observation in CA3. The royal we is Alex Tomei and Daniel Gray in CA3 of young and old rhesus macaques. So in the same monkeys in which we found elevated CA3 firing rates, we also found a decline in a specific class of interneuron, those containing the calcium binding protein somatostatin, but there was no change in numbers of GABAergic of parvalbumin containing GABAergic neurons. So this decline in somatostatin containing interneurons combined with was correlated with both an elevation of the firing rates and performance of a hippocampus dependent task in these monkeys. So what I find particularly compelling about these data is that not only in rats, but also in monkeys, we see a selective anatomical change in GABAergic cells in CA3 and increased excitability in aging. Furthermore, Mark Yassa, among others, has observed increased activity by MRI in aging humans. And so I like to look at this as potentially reflecting a clear species convergence, indicating that CA3 excitability is a consistent biomarker of aging. So I want to move on to synaptic plasticity. We've been over the connectivity part. So the second critical category of change in the aging brain is plasticity at the aging synapse. And I'm only going to discuss NMDA-dependent forms of plasticity here, or we'd be here another 2 hours. But first of all, LTP is harder to induce in CA1 of old rats if you use weak stimulation. But if you use robust stimulation parameters, induction is fine. There's no age difference, not in the dentate gyrus CA3 or CA1. But LTP decays faster in all three regions, and the faster decay of LTP in both the dentate gyrus and in CA1 is correlated with spatial memory performance on the Barnes maze. Furthermore, LTD, or long-term depression that weakens synaptic strength, is easier to induce at CA1 synapses in the stratum radiatum, but unfortunately, here we go again, more complexity. LTD is impaired at CA3 commercial association synapses and stratum radiatum in aged rats. Okay, so now I want to show you some relatively new data just published in cerebral cortex from the monkey. And the person leading this was Daniel Gray, who's now in Lindsay DeHaze's lab. And it's really speaks to some of the mechanisms that may contribute to altered synapse health and result in connectivity or plasticity changes in the aging brain. Now, of course, I don't need to say to this audience, it's clear that the innate immune system can regulate synapse function and integrity through a number of mechanisms. And one of these, of course, is via microglial regulation of the brain extracellular matrix. Now, microglia are thought to regulate the extracellular matrix by releasing matrix degrading enzymes that reduce perineuronal net density around synapses. So the matrix is a complex combination or network of proteins and sugars that can affect multiple synapses, not only neurons, but glia and vascular cells, providing these cells with structural and biochemical support. And the perineuronal nets are specialized extracellular matrix structures that surround some somas and dendrites of neurons. And one prominent cell type associated with perineuronal nets is the parvalbumin-containing GABAergic interneuron. And you see these interneurons as red in this picture, and some of the PV neurons here are surrounded by perineuronal nets, which are in green. And this is in the macaque retrosplenial cortex. To the right, you see aggrecan, a prominent protein contained in perineuronal nets being found in regions of around spines, and below is shown, aggrecan is shown to be along with postsynaptic protein Homer 1. Now, the experiment that Daniel Gray and Salma Khattab conducted in my lab sought to determine the relationship between age, microglia, and perineuronal net structure in cognitively characterized young and old rhesus macaques. And several cognitive tests were administered to these monkeys, and I'm only showing you the delayed non-matching to sample object recognition memory task here. You can see that the older monkeys took more trials before reaching criterion, and they showed more degraded performance across increasing delays. And you see in this figure, the region of the retrosplenial cortex that Daniel and Salma examined. And retrosplenial cortex has been implicated in a number of aspects of cognition, including episodic memory recall, navigation, imagination, and planning. And to the right, you see the markers of parvalbumin and of PNNs that we examined, aggrecan and WFA. And to the left below, you see that there's a reduced number of perineuronal nets in the older monkeys surrounding parvalbumin neurons, interneurons. And to the right, you see that the monkeys with fewer perineuronal nets tended to perform more poorly than those animals with more nets. With respect to microglia, you see examples of microglia in young and older monkeys using the IB1A marker. And as you see here, in this case, there's a significantly higher glial density in the older animals than the young monkeys. And this increased microglia density were accompanied by decreases in ramification of the glia cells characteristic of pro-inflammatory phenotypes. And to the right, you see that older monkeys with more microglia perform more poorly on this recognition memory task. And this relationship did not exist, was not significant in younger animals. Additionally, the older monkeys with more microglia also had lower proportion of parvalbumin neurons surrounded by perineuronal nets. So when Daniel did stepwise regression analyses, it indicated that ACAN or aggrecan densities provided further predictive power when evaluating the relationship between age, microglia density, and this object-based memory function, so that increased age was associated with increases in microglia density, and the increased microglia density probably most likely results in a release of these net degrading enzymes in response to neural activity. So the adjusted R square here value suggested that aggrecan containing perineuronal net densities may be a useful and cognitively relevant predictor of circuit function, since they improve the model fit and compared to using only age or glia density alone. Now, you might be sort of scratching your head and said, trying to put this together, and it is a little backwards in interpretation, but these net structures are generally thought to restrict structural plasticity because increases in these nets are associated with the closing of the developmental period, and blocking them can reopen these critical periods. But one hypothesis that emerges from the present data is that reductions in perineuronal nets in the retrosplenial cortex of these aged monkeys may paradoxically result in excessive plasticity in the old animals. That is, fewer nets, more plastic modifications possible. And how this specifically impacts cognition is unknown, but it's possible that this could allow inappropriate synaptic modification to occur and potentially hyperexcitability, which would disrupt network function. Okay, so I want to continue the theme of this talk that illustrates the specificity of the changes that occur in the aging brain in different parts of the hippocampus. And next, I'm going to give you two examples of age-related changes in behavior-driven hippocampal networks in CA1 and CA3 and cell ensemble recordings in young and old animals. The first for CA1. In this slide, I'm just showing you a few of the simultaneously recorded place cells in a young rat on this triangular track. Each cell is a different color, each dot is a spike, and you see the place fields of each of these different cells, place cells on the track. These two panels are illustrative of place cell recordings from both a young rat on two different sessions in the same day. And what you see is that the same hippocampus map is retrieved on the second session of the day as on the first. That is, the yellow cell still firing here, the blue cell here, the red cell here. So the young rats retrieve the same map from session to session, okay? But on about, most of the time, actually, old rats also retrieve the same map, but about a third of the time for any given old rat, a completely different place field distribution appears when the old rat goes back into the same room, as though the old rat had retrieved the wrong map. You see here that the blue cell is now firing over here, and this orange cell didn't fire at all in the first session. So for CA1, when environments are familiar and identical, cell ensembles in old rats occasionally remap when they should not. Now, I want to show you what happens in CA3. And these are data from Ian Wilson's lab from young and old rats. And in this case, Ian ran the rats first in a circular environment, and then a square, and then back in a circle. And when the environment is changed, like here, young rats show what's called global remapping. That is, the representation in the square is different from the cell firing in the circle. So they show different maps for the two different environments. That's what happens in young rats. For old rats, they on some occasions, they don't remap when they should, as they appear to reuse an inappropriate map, as in this example. And so this is clearly a different kind of retrieval error in CA3 networks of older rats than was observed in CA1. So in CA1, the cells, I don't know what's happening here. Something, but in CA1, cells remap when they shouldn't. In CA3, the cells don't remap when they should, suggesting that the dynamic processes of pattern separation and pattern completion are altered in old hippocampal networks, right? So at the very least, this indicates to me that integrating episodes into the context in which they're experienced is altered or will be more difficult for the older animals. Let's see if I can get rid of that. No, something is. Thank you. Okay, I want to show you now is a new direction that we're going in our aging rat experiments currently with respect to interrogating navigation circuits in the aging brain. We're recording from CA1 and medial entorhinal cortex of young and old rats. And it's clear that these structures are critical for implementing allocentric and egocentric strategies for successful navigation. And so we developed a task to determine the extent to which young and aged animals differ in the use of these strategies to solve this goal navigation task. So this task can put visual and path integration cues into direct and immediate conflict without interrupting ongoing behavior, like taking the animal out of the apparatus to change the cue configuration. And Adam Lester is the person who constructed this instantaneous cue rotation arena that you see diagrammed here. So there's a circular platform inside a track that the rat runs around to stop for food reward in a particular spot, specified by visual images that are projected onto the arena from these external cameras outside the apparatus. Here's the actual apparatus, the arena, and Adam holding the robot that he had to build to completely control for olfactory compounds on the track. The robot only delivers food when the animal stops at a certain location for a particular period of time. And we have behavioral data that shows that both young and old animals can learn this task in a reasonable amount of time. And the experiments that we're embarking on include obtaining more behavioral results of this instantaneous cue rotation for these young and old rats, as well as to monitor short time scale network dynamics in the medial entorhinal cortex and hippocampus when the switch occurs. So we think that with this method, we can probably determine what strategy each rat is using to solve the goal navigation task. And this will enable us to determine how rapidly the networks settle back into a stable configuration following rotation, and whether that's different in aging animals. As I described earlier, we know that spatial navigation is impaired during aging in all species tested from rodents to primates, but it's rare to be able to compare rodents and primates under similar behavioral conditions. And I want to give you two examples where we're at least trying to do just that. One that involves FDG PET, microPET, using whole brain imaging of glucose metabolism to gauge which regions are active during different behaviors. And the other is ensemble telemetered recordings in the posterior hippocampus of the freely moving macaque. Okay, so beginning with PET, we used PET to capture whole brain activation patterns in 4 distinct spatial behaviors in young and old monkeys. And the primary person collecting these data was James Engel. So immediately after tracer injection, each animal was given one of these 4 behaviors before being taken to the PET scanner within an hour. And each animal served as their own control because every animal was scanned under all 4 behavior conditions. So first is just the cage freely moving in a small constrained space. The second is head restrained and moved along a long enclosure. So we have in a primate restraint chair. So there's optic flow, but there's no locomotion. The third is walking in a treadmill. So there is locomotion, walking in place, but no optic flow. And then the fourth is freely moving back and forth end to end on the 6-meter track. Eric Ryman and Kwai Chen have helped us with analytic approaches for these data, and the story is complicated, of course, but I'll extract the major points for you here, and that is that the right posterior hippocampus was particularly engaged in this walk condition in both age groups, actually. And although similar regions across the brain were co-activated in the young and old animals, the older animals displayed network coactivity patterns that only weakly discriminated between the 4 behaviors, whereas the younger animals, the network patterns were much more distinct. And in some cases, in fact, the older monkeys showed a different set of functional connections between regions of interest. So the lack of well-differentiated behavioral networks and corresponding changes in functional connectivity has certainly been observed before in aging humans. And it's consistent with the idea that altered network dynamics could be an important source of age-related changes in spatial cognition. So these data suggested, at least to me, that if we're going to find place cells in a monkey, we need to get our electrodes into the right posterior hippocampus, equivalent to the dorsal hippocampus in the rat. So the other approach we're using to create conditions where behavioral states are similar between rodents is to conduct telemetered recording studies in animals that are completely free to behave without any kind of restraint. And as you know, most non-human primate studies of the medial temporal lobe or any study is usually in a restrained condition. There's one exception. And after years of trying to implement these methods, they are working. And we have implanted tetrads into the right posterior hippocampus to record from these same 4 behaviors that I showed you in the PET study. So we've been able to isolate hippocampal cells with our tetrads, as you see here, and collect single low frequency field potential recordings, local field potential recordings. And so Drew Moore originally helped initiate these recordings, and you see example from him here. And again, this is the 6-meter track. Interestingly, you know how beautiful sinusoidal waves happen when rats or mice run. The theta in monkeys is much more fragmented than in the rat. But regardless of if there's beautiful theta or not, are there place fields? Colin Kyle has done the analysis that I show you here. You see 3 hippocampal cells from 3 different track walking sessions in the 6-meter track, and these are occupancy normalized rate maps with the hotter colors reflecting higher firing rates. Now, the polar plots on the right indicate that actually these cells were active uniformly in all directions, these particular cells. So the short answer to the question of whether the monkey has place cells under these behavioral conditions is that we don't know yet. But we, after the years of being shut down because of the pandemic, we are back. I have a surgery date in August, and we're getting our electrodes into the hippocampus of the monkey that's ready to go. And maybe I'll be able to tell you in some, some lifetime that's reasonable of what the answer is. And we may have an age comparison here as well. So to transition now back to different hippocampal subregions and how each is affected by age. Some of you might have noticed, those who are particularly fond of the dentate drivers or granule cells, that I haven't mentioned those cells yet. And you might ask why. There hasn't been any electrophysiological recording comparing freely behaving animals with granule cells during aging. And yeah, you can ask why. And really, my answer to you is that they are the most quiet, elusive, frustrating cells to record from in the brain. It's really a challenge. I can tell you about our attempts some other time, but in the next part of my talk, I'm going to tell you about methods that I've used to try to get at how aging may impact granule cell networks, if for no other reason to give granule cells some more airtime in this presentation. So I'll discuss with you 2 approaches that I've taken, one using imaging methods in young and old monkeys, the other using immediate early gene activity markers in young and old rats. And to begin this story, we collected structural MRI scans for monkeys equivalent in age to 27 to 87 human years, all behaviorally characterized animals. And the younger animals had better memory than did the older animals. And we found, however, in this study, the total volume of the hippocampus differed by less than 6% between these age groups. And not surprisingly, there wasn't any relationship between hippocampus volume and memory in the old animals. So in monkeys, hippocampus shrinkage can't account for memory impairments. And I think this is an important observation because, as you know, only humans show the classic Alzheimer's disease neuropathological pattern. So we're looking at a living animal who, by definition, is aging normally. So these same animals, however, were also imaged using an MRI technique that Scott Small helped us with that relies on measures of cerebral blood volume that reflects basal or resting levels of metabolism. And this enabled us to compare activity patterns across temporal lobe subregions and in young and old monkeys. And again, these were the same animals that showed no hippocampus volume change. Scott found no correlation between age and metabolic activity in the entorhinal cortex, CA1, or in the subiculum, but a significant negative correlation between age and CBV activity in the dentate gyrus, with older animals showing, older animals showing lower activity levels in the dentate gyrus. Importantly, also, there was a region selective significant correlation between this metabolic measure in the dentate gyrus and memory performance of the older animals. So the more activity, the better the memory was in the older animals. So this suggests then that the dentate gyrus may be particularly vulnerable in normal aging. But of course, MRI measurements don't have single cell resolution and are only an indirect measure of selectivity, but a method that does have single cell resolution and can reflect behaviorally driven activity in cells across wide regions of the brain is a method that John Guzowski developed when he was a postdoc in my lab, this ARC-CAFISH method, which is capable of distinguishing multiple experiences in the same animals across wide regions of the brain at cellular resolution. So John showed that the cellular compartmentalization of ARC mRNA varies as a function of time from the activity that elicited it. And the nucleus shown here is in blue and ARC is in red. If ARC is in the nucleus, then the behavior that activated it occurred 5 minutes previously. If ARC is in the cytoplasm, then the behavior that activated it occurred 30 minutes prior, and you can give two behaviors separated by 20 minutes. And if it's the same behavior and the same cells participated in each, then ARC is in both the nucleus and cytoplasm, as you see here. So just to give you a brief idea of what happens in aging in the different hippocampus subregions, when you have 2 5-minute exploratory behaviors separated by 20 minutes, Monica Chawla showed that young, middle-aged, and old rats show the same proportions of CA1 and CA3 pyramidal cells activated. However, aged rats show a lower proportion of cells activated by behavior as you go from young, middle-aged to old, suggesting that as we saw with the CBV activity for the old monkeys, the dentate gyrus in rats is a hippocampus subregion susceptible to the effects of advancing age. So we think now that transcriptional repression mechanisms involving methylation status of the ARC gene may play a role in preventing some granule cells from being active. That is, Marcia Penner found that there was a greater methylation of the ARC gene in the promoter region within granule cells of old rats, suggesting that these cells may be held in a state less conducive for facilitation of ARC transcription. So these granule cell data then from rats, the activity marker data in monkeys, and the cell recording data in monkeys and rats suggest that the hippocampal circuits that show the most change during aging involve the dentate gyrus hilar region and C3, with reduced circuit participation and metabolic activity in the dentate gyrus and increased excitability in C3 that I described for you in the rat and monkey studies. I want to mention here that this is I'm going to end with this because I think I'm long. This is a completely different pattern to the networks known to be targeted by Alzheimer's disease. That is, in Alzheimer's disease, it's the CA1 cells that are particularly vulnerable to change and degeneration, while the granule cells, for example, do not show changes beyond age-matched controls until very, very late in the disease. So this suggests to me that favors the idea that while normative aging and Alzheimer's can coexist, they show a different signature, a different pattern, and can be distinguished. So I'll end with a summary that I hope what you can take away from this is that there's remarkable selectivity of the changes observed in the aging brain. Another point that this raises is the challenge of discovering one pathway or mechanism that can be used to optimize circuits across the entire brain during aging. The solutions are going to be multiple and complex. Another point is that not all changes in the hippocampus are necessarily detrimental. And there are changes that appear to be adaptive and may keep circuit function stable. And it's going to be important to distinguish these potentially compensatory changes from detrimental changes when developing therapeutics. Clearly, we've learned a lot over the past 4 decades about the impact of age on cognition and the circuits responsible for these behaviors, but there's much more to be discovered. The hope going forward, of course, is that we will be able to figure out ways to optimize cognitive health throughout life. We're working really hard to get there. But I'm sure many people in this audience could contribute to the body of data that will get us there. And thanks. All my collaborators and my funding sources are listed here.
Thank you for that beautiful talk, Carol.